Welcome everyone. Today I will discuss about pathogenesis of bacterial diseases. So what is pathogenesis? Pathogenesis is a collective events that lead to development of disease. A series of events that lead to development of disease is called pathogenesis. And pathogenicity is the ability of microorganisms to develop disease. Now first come to the steps of pathogenesis. First, transmission of organism from external source. Then, evasion of host defense. After transmission, there will be evasion of host defense. Then, adherence to host surface or adherence to mucous membrane. Then, colonization of microorganism. Then, disease production by toxin production or by invasion. Then, host immune response occur. Then, disease progression occur or resolution of disease occur. So these are the steps of pathogenesis. First, transmission from external source, that is transmission of microorganism. Then evasion of host defense, adherence to host cell surface or adherence to mucous membrane. Then colonization of microorganism, disease production by toxin or invasion. Then host immune response occur, and ultimately disease may progress or there may be resolution. Now come to virulence factors of bacteria. Number 1, factors for adherence. These are pili, glycocalyx, capsular polysaccharides, and some other adhesive molecules. These are factors responsible for adherence. Then number 2, invasiveness. Factors responsible for invasiveness are hyaluronidase. This is the enzyme that breaks down hyaluronic acid, which is ground substance, and helps in spreading of infection. Then collagenase. This collagenase enzyme breaks down collagen and helps in invasion. Then another enzyme, immunoglobulin A protease. This immunoglobulin A protease break down secretory immunoglobulin and helps in pathogenesis of bacteria. Immunoglobulin A producing bacteria are Neisseria gonorrhoeae, Neisseria meningitidis, Haemophilus influenzae, Streptococcus pneumoniae. So these bacteria produce immunoglobulin A protease enzyme. Neisseria meningitidis, Neisseria gonorrhoeae, Haemophilus influenzae, Streptococcus pneumoniae. And there is another enzyme named coagulase. This coagulase enzyme prevent phagocytosis. Then come to anti-phagocytic factors. Anti-phagocytic factors are capsule, M protein, protein A. Then intracellular survival. Some bacteria like Mycobacteria, Listeria, Brucella, Legionella, they survive intracellularly. And mechanism behind this, number one, inhibition of fusion of phagosome with lysosome. Inhibition of fusion of phagosome with lysosome. So phagosome cannot fuse with lysosome and lysosomal enzyme cannot destroy the bacteria. This is one mechanism. Another mechanism is inhibition of acidification of phagosome. Inhibition of acidification of phagosome. For action of lysosomal enzymes, there must be acidification of phagosome. So inhibition of acidification leads to inactivation of lysosomal enzymes. And number 3, escape of organism from phagosome to cytoplasm. Escape of organism from phagosome to cytoplasm, it also helps in intracellular survival of bacteria. Then come to toxins. Toxins are two types, exotoxins and endotoxins. I will discuss details about exotoxins and endotoxins. Exotoxins are proteins or polypeptide in nature. Exotoxins are proteins or polypeptide in nature. They are actively secreted from both gram-positive and gram-negative bacteria. Actively secreted from both gram-positive and gram-negative bacteria. They are highly toxic highly antigenic. They are highly toxic, highly antigenic. Thus, as they are highly antigenic, they are used for vaccine production. And vaccines are always heat sensitive, so these exotoxins are heat labile. So exotoxins are protein or polypeptide in nature, actively secreted by both gram-positive and gram-negative bacteria, highly toxic, highly antigenic. As they are highly antigenic, they are used for vaccine production, and vaccine must maintain a specific temperature. That means vaccines are heat labile, so exotoxins are also heat labile. So there are example of exotoxin producing bacteria. Gram-positive bacteria are Bacillus anthracis that produce anthrax toxin, Clostridium tetani produce tetanospasmin and tetanolysine, Clostridium botulinum produce botulinum toxin, Clostridium perfringens produce alpha toxin and theta toxin, which is responsible for gas gangrene. Corynebacterium diphtheria produce diphtheria toxin. E. coli produce verotoxin and enterotoxin. Vibrio also produce enterotoxin. So these are some examples of exotoxin producing bacteria. Now come to endotoxin, and from here we can learn difference between exotoxin and endotoxin. Endotoxins are lipopolysaccharide in nature. Endotoxins are lipopolysaccharide in nature, and it is integral part of cell wall of gram-negative bacteria. Endotoxins are lipopolysaccharide in nature, that is integral part of cell wall of gram-negative bacteria, and liberated only after bacterial death. Liberated only after bacterial death or disruption of this cell wall. So endotoxins are lipopolysaccharide in nature, which is integral part of cell wall of gram-negative bacteria. That means liberated from gram-negative bacteria only after bacterial death. Endotoxins are less antigenic, less toxic. As less antigenic, no vaccine production, and they are heat stable. They are heat stable. So these are difference between exotoxins and endotoxins. Now come to systemic effect of endotoxin. This endotoxin activate macrophage, activate complement and also activate tissue factor. So endotoxin activate macrophage, activate alternate pathway of complement and also activate tissue factor. After activation, macrophage produce cytokines like interleukin 1, interleukin 6, tumor necrosis factor alpha and nitric oxide. This interleukin 1 and 6 responsible for fever. Tumor necrosis factor alpha causes fever and hypotension. Nitric oxide also cause vasodilation and hypotension. So these are effects of activated macrophage, and after complement activation there will be production of anaphylatoxin. So anaphylatoxins are C3A, C4A and C5A. After complement activation there will be production of C3A, C4A, C5A. These are anaphylatoxins, and these anaphylatoxins cause hypotension, edema. And after complement activation there will be also production of chemotactic factors. Chemotactic factor is C5A. This chemotactic factor cause chemotaxis of neutrophil. This C5A acts as chemotactic factor and cause accumulation of neutrophil. And tissue factor activate coagulation system and ultimately develop DIC. Tissue factor activate coagulation system and develop DIC. DIC is disseminated intravascular coagulation. Due to disseminated intravascular coagulation, there will be thrombus formation, ischemia, and ultimately develop multiple organ failure. So these are systemic effects of endotoxins. Now come to examples of endotoxin producing bacteria. E. coli, Klebsiella, Salmonella, Shigella, Proteus, Vibrio, Pseudomonas. So these are some examples of endotoxin producing bacteria. Now I will discuss about Koch postulates. In 1884, Robert Koch proposed some postulates that link microorganism to disease, and you can memorize 4 Koch postulates by 4 words. Number 1 is association. Number 2, culture. Number 3, inoculation. Number 4, isolation. So for memorizing Koch postulates, first memorize these 4 words. The first postulate is microorganism should be associated with all disease in question. That means microorganism should be associated with every disease in question. Number 2, microorganism should be grown in pure culture. Organism should be grown in pure culture in vitro. Pure culture in vitro for several generations. Number 3, when this pure culture is inoculated in susceptible animal, the same disease must occur. That means when this bacteria from culture is introduced into susceptible animal, it will produce the same disease. And number 4, the same organism must be again isolated from that experimental animal. So first, microorganism should be associated with every disease in question, must be associated with all disease in question. Number 2, microorganism should be grown in pure culture in vitro for several generations. Number 3, when this pure culture is inoculated in a susceptible animal, same disease must occur. Number 4, same organism must be isolated again from that experimental animal. So these are 4 Koch postulates, and there are some limitations of these Koch postulates. So what are the limitations? Number 1, individuals exposed to a specific organism not always produce disease. For example, in case of Mycobacterium tuberculosis, in 95% cases, this Mycobacterium tuberculosis doesn't cause disease in individuals who are exposed to this bacteria. So this cannot be explained by this Koch postulate. Number 2, all bacteria don't grow in artificial culture media. For example, Treponema pallidum, Mycobacterium leprae, these bacteria cannot be cultivated in artificial media. Then number 3, some bacteria cannot produce disease in experimental animal. For example, Neisseria gonorrhoeae cannot produce disease in experimental animal. Number 4, sometimes infection may be caused by mixed organism. Koch postulate cannot explain this mixed infection. Number 5, allergic and autoimmune diseases cannot be explained by this Koch postulate. And number 6, there are some ethical problems. That means introduction of microorganism into healthy individual, what is said in Koch postulates, it is totally unethical. So these are limitations of Koch postulates. Now come to molecular Koch postulate. Molecular Koch postulate. First, phenotype or property under investigation must be associated with pathogenic strain, not with non-pathogenic strains. Phenotype or property under investigation must be associated with pathogenic strain, not with non-pathogenic strain. That means only pathogenic strain is associated with pathogenicity. Number 2, a specific inactivation of pathogenic gene, a specific inactivation of pathogenic gene leads to decreased pathogenicity. That means when pathogenic gene is inactivated, pathogenicity will be decreased. And number 3, replacement of mutated gene with wild type of gene leads to restoration of pathogenicity. That means when mutated gene is again replaced by wild gene, then again there will be restoration of pathogenicity. This is molecular postulate. Now today's last topic is biofilm. Biofilm is a protective matrix. Biofilm is a protective matrix produced by some bacteria which protect bacteria from antibiotic and also protect from host immune response. So biofilm is a protective matrix produced by some bacteria which protect bacteria from toxic effect of antibiotic and also protect from host immune response. Now come to composition of biofilm. It is composed of some proteins and polysaccharides. Now come to examples of biofilm producing bacteria. Number 1, Pseudomonas aeruginosa infection in cystic fibrosis patient. Pseudomonas aeruginosa infection in cystic fibrosis patient. Here, Pseudomonas aeruginosa produce biofilm. Number 2, Staphylococcus epidermidis infection in prosthetic heart valve and joint prosthesis. Staphylococcus epidermidis infection in prosthetic heart valve, joint prosthesis, dental plaque. Here, this Staphylococcus epidermidis also produce biofilm. And importance of biofilm is this biofilm protect bacteria from lethal effect of antibiotic and also protect bacteria from host immune response and thus contribute to chronic infection and increases morbidity and mortality. So this is end of today's session. If you understand my class, please like and subscribe for more videos. Thank you.